Now this video is about what is a gene. We've talked about that many times, and of course I told you that on a chromosome you have small pieces of DNA that attract the RNA polymerase. Those are of course called promoters, and here is our data. That is the sequence of chemicals here that attract the RNA polymerase. The RNA polymerase of course is the copy machine. It's attracted to the data. That's where it will attach, and then it will start copying the DNA in the form of an mRNA. The mRNA is a copy, and I have another video that talks more about the copy and how that works. Right now, we just want to talk about how this structure forms a gene. Of course, we have a promoter here, and the promoter, whatever sequences, there are many sequences, not just TATA, called promoters that attract RNA polymerase. So this protein called RNA polymerase, it's this red ball here, it will be attracted to specifically these special sequences. And wherever these special sequences exist in a chromosome, you're going to get RNA polymerase attaching. And when it does, it's going to start reading or moving, essentially, if you want to think of it that way, reading, but moving across these letters. And it's going to assemble a copy. Now that copy is going to be very similar to this complementary piece down here, which I have another video to talk about that. And it's going to move across this region, and it would continue making a copy, thousands, tens of thousands, hundreds of thousands of letters here in this green one, this mRNA here. It would continue making a copy until it runs into something that's going to cause it to fall off the DNA. And in this particular case, the thing that makes it fall off is the terminator. And the terminator has to do with changing the shape of the DNA. Realize that actually when this is going on, when this attaches, these two pieces of DNA, these two single strands, have actually kind of become loose. They're not binding to each other very well in this region, and that's actually what allows the promoter to stick. I mean, the RNA polymerase to stick to the promoter. Well, that means this is kind of like single-stranded and kind of like a sheet floating in the wind. And when it does this, when this is partially, at least the region here is partially single-stranded, these bonds have broken for some reason. The A's and T's here, notice that there are a bunch of T's right here, and there are a bunch of A's right here with some G's and C's in the middle. Well, these A's here and these T's here can stick to one another, and they form that thing that I described in class called a hairpin loop. Well, that, of course, is not tenable, and that causes this to break off. Actually, believe it or not, it's really not the DNA that forms the hairpin. Not that the DNA can't form a hairpin, but actually the RNA polymerase moves actually into the terminator, and the RNA itself forms the hairpin that actually makes the RNA polymerase come off. But we don't need to know that. We just need to know that there are some certain letters. Whenever you see these letters right here, where you get a bunch of T's followed by some A's, or maybe some A's and T's with some GC's in the middle, actually any sequence that can bind with a sequence nearby will form a little hairpin. And that's a signal that that's probably a terminator. So the idea here is there are many types of terminators. I'm giving you the simplest one here, where it's all the same letter, then some other letter, and then all of a letter that can bind to these letters up here in the front. So you have this region binding to this region. And this would be like the simplest terminator. So the idea here is this promoter here is where the RNA polymerase, the copy machine, can stick, and that means it's telling it this is where my gene is. My gene is going to start here, and my gene is going to be anything in between the promoter and whatever causes the RNA polymerase to stop copying, which means fall off the DNA. And so this attracts it, so this determines the start of the gene. And the terminator, which makes the RNA polymerase fall off, tells you where the end of the gene is. So the idea here is we have a gene here, and it's defined by the fact that we have a TATA here, and we have a terminator here. So the region in between is copied in the form of this mRNA called a messenger RNA, and this green piece right here, the completed mRNA, is our copy that we're going to send out of the nucleus to the ribosome. So this defines what a gene is. A gene is a region of DNA where the RNA polymerase can stick, called a promoter, and copy the region adjacent to it. And that's what we're seeing here in this video. As it moves along here, it's making a copy. And then it'll hit a region that's going to cause it to fall off. That's going to be the end of that gene. Now, once this process occurs, as we have in this video, you actually, the mRNA is separate and it's going to leave. The RNA polymerase is also free to leave, and the polymerase could loop back around over here and start making another copy, or it could go on to another gene and another RNA polymerase. You have many of these RNA polymerases just floating around, and as soon as there's an opening, it's going to stick and it's going to make a copy. All right. Now, later on, we will talk about things that can slow down the RNA polymerase, or maybe even make it so that it can't attach at all, in the form of histones, and how tightly they wrap around, or the DNA wraps around them. We'll talk about that in a different video. But this region before the promoter is also important. It regulates the speed and whether or not the RNA polymerase can attach. Some things, other proteins other than RNA polymerase, are going to attach up here in this region right before the promoter. And this region will do what we call regulate the RNA polymerase, or regulate the gene. And what it's going to do is it's going to basically say, how many of these copies are we going to make? When certain things stick up here, it might speed this up, or it might slow it down. And then other things can happen. We talked about methylation, which I will have another video, which will basically shut it down completely, so that this gene is not made at all.